Hello. This is a podcast, and it is not for the children's. It is a podcast. Did you like that?
Children's is already plural.
Just want to make sure. This is a podcast where four friends get together, try to amaze and astound each other with facts about the world enough that one of the other people on the podcast might say, "Holy fucking science." Hello, I'm Stefan Chin. I am now Hank Green. Or I'm just hosting this podcast for today. It's actually that one. Okay, so
It's going
I don't
well.
know. That was good. I've really done a number on this podcast. I'm Stefan Chin. I'm a producer and host on the semi-popular YouTube channel SciShow.
Moderately successful.
Today we're joined by Brit Garner. How you doing?
I want to be here right now. I'm
this
glad
is
I'm
the
here.
place to be.
Yeah.
We're doing good. Hi. Okay. We're also joined by Tuna. Hi.
Question mark as in what's your last name? Is that what that was?
Are you Tuna on the podcast also?
Yeah, I think so.
Okay.
That's just
Beyonce, just
Just
one
Tuna.
word.
right, yeah. I just like,
Cool.
It's going to be a symbol in a couple months.
yeah. If you need the last name, it means I don't count yet.
Mm.
Oh.
Yeah.
Oh, shoot.
I know.
That's deep.
Yeah.
Maybe. Tuna works on SciShow also, and SciShow Psych, of which Brit hosts. We're making all the connections here.
Yeah.
I do that. Tuna's the one that does things like makes graphics of me riding a shark or inaccurately putting me in Ravenclaw.
I tried.
Wait, you're not a Ravenclaw?
We're not going
she's
there.
Hufflepuff.
No.
I have learned this.
She's
That's
a Griffin.
still not right.
Is it still not right? You spent the whole man. All right.
I love you for your effort.
Are there half houses? Can you be a Gryffindor?
You can be, yeah, or
I
like
say
a
yes.
Slitherpuff.
Well,
Slitherpuff.
I'm
But we're
going to
gonna
tell
get
you,
derailed very
there's about
quickly, friends.
17 different Harry Potter wikis out there, and I
The
researched
fact
them all
that
and
he
still
went to
failed.
all of them is why I forgave him as a human.
But if you want to find out about half houses, I'm sure at least one of the 17 has like a stub
I
on
just
it.
don't know how you fail. Like you're in a house of some kind.
Yeah, but I picked the wrong one because I am apparently a really shitty hat.
Okay. Tuna.
You're a
Bad
beautiful
hat.
human.
Bad hat.
Bad hat,
Joined
beautiful
today
human.
by Tuna, the shitty hat. And lastly, we're joined by Ceri Riley.
Hi.
How's it going?
This is good. This is a good group
This is
of
a good-
people.
She's like,
Yeah.
is it too late to leave?
Yeah. I came for the
Last
science,
chance.
not for the friendship. Excuse me.
There's not going to be too much of that tonight.
Oh, yikes.
Well, actually, Brit's pretty scientific.
Brit's very scientific.
Yeah, take it back. I value my friendship as much as my science.
Yeah. I do too. I think. yeah, it's
That's
good.
a weird thing to think about. I don't feel like comparing your value of friendship and science. Like what?
The amount that I value them in my life are equal. Not the thing itself, but my valuing of the nouns.
Mhm. Like my love that goes into the friendship pool and science pool.
Huh.
Just everywhere.
Okay.
Like I love both of those things equally, which is why I'm such good friends with science.
Speaking of science, this is a science podcast.
Thank
Today we're
God.
going to be talking about miniatures. So like miniature replicas, models, little toys, D&D miniatures, maybe. Maybe there's some science there. I don't know. Brit's saying no.
Psychology for sure.
What does that mean? I feel offended for some reason.
No, don't.
So to decide who goes first today,
Oh boy.
I wanted to do it by who has most recently played miniature golf.
Ooh.
Does that exist here?
I have no idea. So I haven't played since coming to Missoula. So I don't actually remember exactly when the last time I played, but it's got to be like 5 or 6 years.
Me, it's years.
I'm
'Cause
gonna
I got
say-
very angry once, and I've had a problem doing it again since. Because I just got frustrated.
It's supposed to be fun.
No, it's not. That's where you're wrong. It's been probably the same amount for me. So do either of you have any more riveting?
I mean, what is the threshold for mini golf? Because I've only played it like twice, and it was like
it literally says it on the sign when you walk in.
But I mean, do you need like the ceramic paper mache monkey on the golf
No,
course,
no,
or like
that's
the windmill?
Just small courses.
The quality
Just
of
small.
the course is not.
Okay, so it's just small. Then it was 2006.
Holy shit! Do you know that to that detail?
Holy.
Yeah.
Because it was the year.
Maybe it's traumatic. Like,
No.
He's like,
No,
it
was just the year after I graduated high school, but before I found new friends. I had one friend that I hung out with, and we went to go play mini golf one time.
Dramatic.
We only went once because it was
different.
bad mini golf.
What did I say?
Dramatic.
I didn't say dramatic.
We'll check in the edits.
Let's play the tapes. Cool. So, Ceri?
I think mine was probably 2011 when I was still in high school.
I was going to say 2012 for mine. Why don't you do 2013 and then you can go first?
No, because that's not factual or correct, and I don't know if
You said it's about the same.
Yeah, but about is not the same
my
as
God.
most recent. I really don't know. It's been years.
All right. Do you want to go second? I'll go first. You go second.
I guess I just wish maybe you'd thought of a better thing before coming to
I like
host
the mini
this.
golf one.
No, I do too. I do too. I just
You're a
wish,
good
I
hang,
wish I had an
Stefan.
actual answer. I wish it wasn't so far back. Yeah.
I was surprised once I thought of it, I was like, wait, when? I can't remember when I played mini golf,
really
which
don't.
seems like I really just we should all four of us go play mini golf right after this.
It just cuts to us.
Yeah.
playing-
Actually, let's just go now.
We'll
I
do
think I was
this
in
live
Florida,
from the
which
course.
would take me to.
They have mini golf in Florida?
Okay. Never mind.
Just one.
Never
Just
mind.
one.
Just the one
It's like
mini.
2010 kind of thing. Like, anyway.
Okay, sure. I don't know. All right.
Neither I.
You're going second.
Great.
Ceri third, Tuna last. Okay.
Okay.
So my thing is about computer chips because they are very, very small. Miniature, you might say.
No, miniature is relative. Can I put that out there for a second?
well,
Because
we're
I quibbled
getting
on
there.
this.
Okay.
A small thing does not make it miniature, right? If it is in its existence, that as itself.
They have definitely gotten a lot smaller.
Okay,
from
but
when they
something
were
progressing to smaller is different than having a miniature X, right?
Okay, but we talk
X
about
having
like
devolved to a smaller form.
this is very
Evolved.
weird.
There are rules to
Isn't
You're
this.
inviting me
Isn't
hard.
that process called miniaturization?
That's what I was gonna say. We
'Cause
talk
that's
about
like-
miniaturization in technology.
Ooh, in technology. I know
Yeah.
not of this. Go ahead.
Computers.
All right, we've convinced Brit that we can.
We
proceed
got
with the
there. We
podcast.
got
That
there.
is the word.
Oh my. Okay. So yeah, I don't know.
Flag on the play.
Fight.
Penalty.
Yeah.
Yeah, I loosely follow like computer tech, but I didn't really realize where we were and like what the plans are for the immediate future. Because like we did an episode on Moore's law. It's not a law. It's just an observation.
Lowercase L.
Okay.
Yeah. Well, it is uppercase.
But Moore thinks
Use
he's way
least-
more important than everybody else thinks Moore is.
Yes, basically, that's 100% true. But it's the observation that the number of transistors on chips doubles every year. And that was his original prediction, and that was revised in the 70s to every 2 years. But that's been pretty steady since 1975 up through 2012.
For us, we talk about Moore's law with genomic data and basically the cost of sequencing. So just the idea that like to sequence 1,000 loci on a individual salmon has just dramatically gone off because of the rapid increase in sequencing ability.
Yeah.
So we see it in biological science as well.
Is that in
With
that
cost and
field
production.
is the rate of growth falling off, or is it pretty steady still?
Not steady, linear, steady. We're still exponential.
Right. Yes.
Yeah.
Yes. But like Moore's law, it's still going. It's just not going quite at that doubling rate.
Mm-hmm. Gotcha. Yeah, I'm not sure because there's the price function and then there's the actual like acquisition of number of data points, and it's looking at the interface of those two, which is what's interesting for things like conservation or like when you're applying or using those things.
Okay.
Man, it sucks that this is an audio podcast for most people because I could really use some charts for this. Like if you could just draw me a diagram.
Brit's got some diagrams.
You have diagrams?
when we get there.
Nice.
Maybe. I peeked on your computer.
He cheated. I said this. I literally said this as a joke, and yet there I am, correct. Cheater.
Sneaky Stefan.
So Moore's law. In 2015, Moore himself actually said that his law was dying sometime in the next decade or so because we're kind of reaching the limits of physics for the transistors as they were originally built. Like we've just been making them smaller, fitting more of them, adding more chips, and like we're getting pretty close to like where we have to change our strategy. So Intel has talked about their plans specifically, which are actually still in line with Moore's law continuing for a while. So the current chips that they're using are 14 nanometers.
Holy fucking science.
Actually, are you being sarcastic at me?
No, I don't know. Tell me if that's significant.
That seems significant to me.
It seems very small.
It's very small. So
there,
for reference.
then yes, I say it because that's very small.
For the children who are listening right now, despite my warning.
They should be.
A nanometer is a billionth of a meter. Ribosomes in your cells, this is for the children, ribosomes.
This children is excited.
This children.
This children. See, singular.
Yep.
Ribosomes are about 20 nanometers. Helium atoms are 0.1 nanometers, so that's smaller, but still, that's like a single atom. Mm-hmm. One thing that kind of blew my mind, because I just didn't really think about it, was that wavelengths of visible light are hundreds of nanometers, like roughly 400 to 700. Mm-hmm. So like these are just incredibly small, and like those current transistors are about 70 atoms of silicon wide, which that was the HFS thing for me, where I was like,
really?
We're measuring it in atoms?
Yeah, that sounds like that's a horrible
Brits.
way to measure it, though, because a silicon atom is going to be different than a hydrogen atom.
Yes.
Like, that means nothing to me, right? When you think about, like,
Well, it's all
electron
relative.
density in clouds and then, like, actual atomic radii, like, that's different trends for the periodic table. That would mean something totally different. So that's a horrible measurement standard.
I think that's why they have the nanometers too, right? Is the 14
Thank God
nanometers
for
is also
nanometers.
is also
much
the
more.
Don't you be telling me you're measuring in silicon atom. You better give me that NM next to it. I'm stressed out by that.
So Intel currently has plans for 10 nanometer, 7 nanometer, and 5 nanometer chips.
Why those?
Those are just, so there's a body called the International Technology Roadmap for Semiconductors. Maybe that's not a body.
Can we get t-shirts? Why is that a body?
A roadmap. I thought it was an organization at first.
It's like a PDF.
It's an organization of roadmaps.
Yeah.
Rand McNally, they just like meet up once a month. That was so dumb. I'm sorry.
Really showing your age there, Brit.
What does that mean?
I feel like Rand McNally is not a thing.
I don't
Is that
know.
still a thing?
It was the
What is?
They do maps.
It was the
I don't know
quickest.
that.
See, Ceri doesn't even know.
It was the quickest name associated with maps I could grab.
Yeah, for
versus
old
like
people.
Cartesian.
Did they just do maps? I thought they did like school textbooks and stuff.
Oh,
Oh,
I might know
McGraw-Hill.
this. McGraw-Hill does Texas,
Oh,
yeah.
McGraw-Hill.
Oh,
Yeah.
What are
that's
we
what I'm
even
thinking of.
talking about anymore?
Oh, man.
Google Maps
Yeah,
by Rand
please use
McNally.
something relevant.
Yeah. When I was small, I used MapQuest. I
Yeah,
never
MapQuest,
used like physical maps.
Which now tells you where all the pizza restaurants are.
Yeah.
That's its only utility.
Yep.
And
Pretty
they measure
much.
their distance in atoms, which is really confusing.
8,000 silicon
I don't
atoms
know.
to the nearest dominoes.
Do you
It's the
deliver
worst.
within a 10,000 silicon atom radius? Because is that a lot? I don't know.
He
very
is a delivery
small.
driver for
I'm right
Domino's.
outside.
Just screaming at them to use nanometers for the love of God.
So on this roadmap,
This body,
this
got
body,
it. Yeah.
they've set like target dates for each of these sizes.
So 10 and 5 makes sense to me. The 7, I mean, not makes sense, but I see them, they relativistically
I'm
make
sure
sense
there's
to
a
each other.
reason for the ratios. I don't know that reason.
That's all I need to know. Thank you for your honesty.
So they're shooting for 5 nanometer chips by 2020. But the 5 nanometer one is kind of the big one because once you get smaller than...
That's the small one, actually.
Because once you go below 7, that's when you start to have quantum tunneling.
Right.
And so there are people who... Well, I guess I should talk about what quantum tunneling is.
For the children.
For the children.
Yes.
Yeah, because clearly we all know exactly what
We're all up on that.
Well, let
Obviously.
Tuna explain it, maybe.
Yeah, go Tuna.
For the kids, Tuna. For the kids.
Yeah.
For the kids.
So
well, you see, there's this TV show called Quantum Leap that was really old,
Ooh.
and people don't know it anymore. No, that's
I barely know quantum.
Brit
okay.
does not like this.
I like your attempt,
Yeah,
but
Was
but
that
you really
produced
hate
by
Scott
Rand
Bakula.
McNally? Oh.
Well, Rand McNally was the
sorry.
guy with the teleporter, yeah.
Can anyone else think of a map maker besides Google?
No, because
I know a lady named Patty who makes maps.
I swear to God. Her and Rand can meet up at this body.
Patty and
I'm trying to defend myself here because here's the thing. I'm not aging myself or dating if the only example or type case or instance is Rand McNally. And I feel like he monopolized the map game. So it's like, you said board games and you're like, oh, like the Parker Brothers. Yeah. I got confused myself. Then you're like, oh, you're old. You're like, no, it's just they're the only ones. I'm done. I'm just very self-conscious about Rand McNally.
Let's not go on a tangent about board games because I have thoughts about that, but we're not going to go there.
I've heard LA has a bustling board game.
It sure does.
This guy who was on my shoot earlier who was telling me that it's really hopping.
Yeah, the board game scene in LA is off the charts.
Wow.
It is nuts. Okay, so quantum tunneling
is a hit new board game.
It's
by Milton Bradley. Oh, that's the other one. Brains are so stupid.
Oh boy.
I'm gonna quit. Go ahead.
So when the transistors, they get too small, there's not enough material between them, and so the electrons that are shooting through these transistors can just jump right through the wall, which is not good when you're trying to transmit information accurately.
It's like myelination when your nerves lose myelin. It's the best biological analog.
Oh, that's good.
So things like MS, I'm pretty sure affects myelin.
Okay.
You're basically removing the ability to
Thank you for that.
Relevance.
Lovely and relatable example.
It's not just tech, living things. It's great.
So once you start getting too small, below the 7 nanometers, that's where the quantum tunneling starts to become a big issue. But apparently they have plans for 5 nanometers, so they think they can do it.
Just because they can, should they?
Yes?
No.
Oh, I'm going to look back at my
if
notes.
they patent it and package it and slap it on a lunchbox, I'll go buy it.
I love you so much right now.
I'm not even sure the point I was making there. But the next level is finding other ways of speeding up the chips. And so some of them are using light instead of electrons, which is kind of cool. And there's one called Vortex. It's like a Vortex laser beam.
What does that mean?
So
it's holy fucking science for that term.
Yeah.
Vortex laser
cool.
beam, TM.
It sounds scary and awesome at the same
So it's
time.
like
it on
a
a
laser that's warped into a helical shape. And then you can stack them, and each helix can carry a different stream of information. I don't get it at all.
Like the laser itself?
Yeah.
Like the
everything and
light?
learn
Yes.
all about that right now.
I guess
the problem.
It's kind of like light, right? Because lasers are just
Yeah.
photons, and so it's like a different way of shaping light to transmit information.
I wish I could tell you, but I don't understand
Stefan,
it.
all we care about now is the Vortex laser.
I'm sucked into the
Okay.
Something to Google when the children go to bed.
Well, I am going to.
Yeah.
So
Okay.
I'll let you know.
Incognito browser.
I'll use
Safe search
incognito.
off. Vortex laser.
Don't get caught Googling this one at work.
Ooh. Dude, if you typed it into ThinkStock, I guarantee you there would be some sexy vortex lasers. They cannot have a stock photograph topic without having someone in a bikini doing something.
This can be a
I
new
don't know
sexy
how.
Vortex costume. Vortex lasers.
Oh my gosh, see you
Sexy
there. Can we
vortex
make one?
laser.
That'd
Yes.
be great.
Oh my
I like
God.
how we're going to share the
We're gonna
costume.
share it. We're gonna be in the same vortex.
It
It's
is a
fine.
helix, so I feel good about our options.
Yeah.
There might be some
Swirling.
something to be done.
So other alternatives are just different materials like graphene. And in the UK, they've actually made a single transistor that was 1 atom thick and 10 atoms wide.
But which atom? Stefan?
Graphene.
Not silicone?
No,
Not helium?
it's graphene.
I must have a really outdated Rand McNally periodic table of the elements because I don't remember graphene.
That sweet, sweet carbon.
I don't know.
It's just a further proof of the fact that we need nanometers, not atoms.
Okay,
As
all
the,
right.
This is for the dominoes issue.
But still,
It's still
any
incredibly
atom,
small.
Any
Oh, yeah.
atom
I give
would be surprising to me.
relative because hydrogen's sitting there like everybody's very large, right? That's how hydrogen sounds.
Sure. I guess I just imagine if you have like one string of atoms, that it'd be kind of floppy.
Now, structural integrity, different story than size. So that's fair. Amazing because of just the integrity of like
Yeah.
a single.
That's the thing to me, is I'm just like, how do we control this? Like, it's amazing to me that we have the ability to manufacture it and then it like works.
Well, our epithelial cells are a lot of times simple epithelium are one cell thick, and they are like things as important as ducts in our bodies, right? That's incredible. Single cell.
That's the human body, though. No one understands that.
Oh, never mind then. Sorry, I was mistaken. I'm just saying amazing things happen in like in our living form that
That's
are
true.
one cell thick.
Sure. A cell is many atoms, though.
Oh boy. Good point. Mini silicone atoms.
True fact.
So the other one that I thought was cool, great name, is chaos-based circuits.
Holy fucking science.
Which, again, I have this one. I have even less idea of how it works. I'm like,
Do you know what the chaos is?
no. I
Oh.
tried to figure it out, and I did not have enough time
no.
I mean,
to figure.
put your
it.
hand like this.
Right,
and then
yeah.
I'm gonna
Put a
do
drop
the-
of water,
Yeah.
and which way does
He's
it
not
roll?
wearing short enough
That's
shorts
like-
or having a nice enough like hair.
It's just a bunch of hands
But then do it
on
a
a
second
circuit,
time.
and you just drip water on, and then they go different ways. And somehow, that's a computer, I guess. I don't know.
Yeah, technology finds a way.
Oh boy.
Oh, this is holy
Iffy.
fucking science.
I guess
here.
if you count like which way it rolls is like a binary 1 or a
Ooh.
0, then if you
And it
have
can
a row
roll
of hands,
so many different directions.
For it to be binary, it's did it go in the same way? Yes or no? Right?
That's true.
Because
Oh my
otherwise,
God, the
Yeah.
possibilities are limitless.
like life, really,
Like life.
like life, I think,
Good.
which finds a way.
Exactly. Yeah, I don't know. That's all I got on computer chips. I just think they're fucking amazing.
They are.
They're very cool.
And I'm astounded that they work. So it makes me say holy fucking science.
Holy fucking
True.
science.
For me, the question is always why smaller. So I was only halfway kidding when I was doing the microcriton just because they can, should they? I mean, truly, what are you getting in return besides being like, did it? You know, which fair enough. Like that's very cool to say. Like I can't freaking believe we got them down to 5 nanometers.
Yeah.
But are you getting better efficiency, lower cost, faster speed? Like,
Yeah.
what's the
and
utility?
I guess I don't know how much more the trend can continue, but the miniaturization of microchips has been linked to like accelerating productivity from 1997 to 2004, which is like could be linked to economic growth.
Sure.
And so that's like important to think about, I guess. But like how much further can it go? I don't know. Like, are we at the point that the devices will be too good and they're just distractions now because we've got Flappy Bird or something? You know what I'm saying? Do you
Am I
feel
supposed to
me?
know what he's talking about?
Did I lose everyone?
What's Flappy Bird?
Oh, I know
Oh,
Flappy
There's a
Bird.
little
It's the
pedal
game
thing.
It's
you
a
tap.
mobile game.
Yeah.
It's like instead of being angry, it got flappy.
Yeah.
Yeah.
Sure,
Much simpler than
sure.
Angry Birds.
So just
Yeah.
adjective bird, and you're like,
You've got a
good to
hit
go.
game. Yeah.
Gotta
Hit game.
Hit us with some magic and diagrams.
Okay, just because you looked over my shoulder and there happened to be a cell in vivo like panel of microscopy does not mean it
I
has
don't know what
anything
that
to do with
means.
that.
I do.
That's just your
That's what I
desktop
paid
background.
thousands of dollars to learn how to know what it means.
Oh, nice.
Yeah,
Yeah.
yeah, it's amazing. They're beautiful pictures.
It's just living
But-
cells, basically.
in vivo, yeah, living and then microscopy, so taking pictures of.
Very small.
what they're doing.
Yeah.
Okay, so this is what happens when we cheat, Stefan. We get the wrong answer because that actually wasn't the thing I was looking
Oh,
up. It was the
no.
supplemental images next to the thing. Have we all learned
Did you-
something?
Did you ever do the thing where like,
Probably.
someone was you knew someone was cheating off of you, and so you wrote the wrong answers, and then after they turned in their test, you erased everything and wrote the right answers? You apparently have some story.
I won't.
Okay.
Oh.
Okay. Or were you the person that was cheating?
I need you to take that back immediately.
Okay. I don't know about your past.
But you know about my honor, Stefan. That shouldn't matter enough. No, I noticed somebody cheating off of my PSAT, which was
Oh.
determinant for,
Yeah.
national merit and those things, but she was in the next row and I knew that they'd given different forms. So once I noticed it occurring, happily let it occur for the rest of it. The poor thing, she didn't get national merit for some reason. I just don't know why. Cheating, it's bad for the children.
For the children. We've got good messages here.
Yeah.
You guys, we really are about the messages.
It was good for me because when people would try to cheat off me, it'd be like, joke's on you, dummy, because I don't know what I'm talking about.
Again,
Right.
wonderful human, terrible hat.
Terrible hat. Not a
You
good
are a
hat.
gem, though. Truly. Yes. So I did some extensive. So we were told miniatures was the theme, which again, I was like, okay, well, are we going noun or adjective? Are we just going for things that are the smaller version? Or are we going for miniatures, right?
I mean, we do have very strict rules here on Holy Fucking Science.
And by that, you mean I literally beg that there be strict rules because I have trouble finding topics otherwise. But then Caitlin's like, it's fine, have fun, smiley face. Love you guys, have a good day. And it's just joyful. And I'm like, but what are the guidelines? So I did some things and found in the last 2 weeks of scientific articles, looked for the phrase mini or miniature and found something quite cool. I would like, if I may, to take you on a strange journey kind of thing, because this goes back to 2013 when the original instance happened. And then since then, amazing things have occurred. So in the magazine Nature in 2013, there was an article titled Cerebral Organoids Model Human Brain Development and Microcephaly. So.
Wait, I got to ask about one of those words. Organoids?
Oh my gosh, Tuna, that's the exact right word to ask about because that's totally what it's about. Right? Because that got a bizarro, I mean, just
It
like.
strikes me as an interesting word.
Right, because oids, like etymologically, we think like something in the semblance of, like a, like.
All right.
I mean,
Viroid.
Yeah, exactly. Like a smaller
okay,
version or
okay.
like android. So like oid. I mean, I don't
Like
know.
a
I think of that
miniature
dude
version.
that tried to steal pizza in the 90s. Was it the 80s? What was it? Avoid the Noid? I don't know which pizza place it was.
Back to pizza. This is great news.
I don't.
The poor kids.
Yeah, I have
the poor
no
kids
idea.
were all
Okay,
hungry.
good. It is not just
Yeah.
me being a baby.
we're just the young ones here.
Well, organoids, cerebral organoids. Yeah, you're right. This is a bizarro thing to say because that OIDS ending is like, well, what's going on here? So human brains, what an amazing thing to come from that same line of cells that make everything else, right? What a complex and insane little piece of machinery. The thing is, of course, studying human brains, you run into a lot of ethical and practical considerations. So like the ethical in terms of testing and practical in terms of working with something so complex we don't understand. So whenever we have that in medicine, a lot of the times we go to models, and models might be either, well, nowadays we can do some really amazing things with computers, but usually animal models, so things that represent, and we can see, oh, well, this is how it works in a rat model. But a lot of those, in fact, almost all of them, I mean, you just simply aren't representing the complexity of the human brain. You just
Right.
aren't. Like something like a heart or maybe vasculature might be a little closer to a one-to-one, you know, like blood is blood, but like a true human brain, the differences there structurally,
I mean,
you know.
I don't know. I had a roommate that I feel like passed pretty well for a
Was it
rat.
Rand McNally?
You guys,
he just kept making maps. Like, what the fuck is wrong with this guy?
There was cheese at the end of every
swear
road.
to God, we're going to get sued by Rand McNally and it's all your fault. Here we go. Here we go again. This is why we can't have nice things. And so basically the big thing that we've been trying to do is like get these in vitro models. And a lot of that comes from the idea of stem cells. Like, can we make a model, but a model that's actually representative instead of mouse, and we're trying to model a human brain. But again, you have the ethics issues and the practical issues. So we have used human embryonic stem cells and two different types. You can have, two different types of stem cells, which are embryonic. We use those, but we also use the ones that can be induced pluripotent. So embryonic are pluripotent on their own, meaning they can turn into what they want, what they will. But we also have ones that are not embryonic that you can induce to be pluripotent. The point being, 2 types of stem cells typically used to become things, become these living tissues. So with the brain, though, it has been trouble because, like, how are we going to actually make a brain, you know?
Sure.
And so this is where this term organoids comes from, which are organ-like structures. And so in 2013, scientists in Austria successfully developed a complex human brain tissue in a 3-dimensional culture system and termed it mini brains. Indeed. And so what this does, cerebral organoids, so cerebral, so cerebellum, brain organoid, i.e., in quotations, mini brains. So they use the term mini brains as a follow-up to cerebral organoids. Not only that, but they actually had distinct regions. So we're not just making neuronal tissue, distinct
it.
regions.
I'm going to give that a holy fucking
I
sign.
don't like
The fact that there's like,
Thank
Yeah.
you.
Yeah,
Thank you, ma'am. Yes.
Are they
Stefan's
alive?
so freaked out.
Are
he
they
can't
alive?
talk. Whoa, buddy. I don't get paid to answer those questions.
did.
That's a whole nother level.
hard
Where-
question.
So,
Brit, where does consciousness come from?
I
Really?
swear to God, I cannot start from chaos theory, clearly. Yeah, so different ethics issues there, right? Just because this is amazing, right? It's saying if you make this, we don't have to deal with people potentially upset about using animals or potentially
Sure.
upset about
Yeah.
using da. Hey, we made a bunch of mini brains. No harm, no foul. We good, right? And that has amazing implications for being able to cheaply and quickly have actual human brain tissue.
I just
Okay.
don't know if we good.
Yeah.
I'm freaked out.
Okay, but don't be. I mean, maybe be. Again,
I want to know when we can
different
power
question.
an iPhone with those suckers.
I mean,
Like the Matrix.
Yeah.
You just have.
you guys are not thinking about this in the right way at all here. We're talking about curing disease and you're wanting to charge your iPhone. This is not
No,
what
the worst
we're
part
doing.
is
They
I
want a
don't
new
even have an
Siri
iPhone.
is what they want.
Yeah, well, no, that's fine.
Just a brain.
Well, quickly, this is kind of the breakdown of how it works. So we have both those 2 types of stem cells, embryonic and induced pluripotent. They wound up being able to generate this layer, which in the human body we'd say a neuroectoderm, neuro, brain, ecto, outside derm, skin. Neuroectoderm, that's usually where nervous tissue comes from. And then they put those little tiny fragments in this 3D culture and like gel so that the proteins could scaffold. And then they saw that after 15 to 20 days, that's when the mini brains were there, which was basically continuous epithelial neurotissue surrounding a fluid-filled cavity. Now that's incredible because our brains, like the ventricles that we have throughout our brains, like they actually saw that after 15 to 20 days. After 20 to 30 days, they actually had defined brain regions, which were cerebral cortex and meninges, 2 of the things that they had.
That's
Yeah.
crazy.
Only
So
4
it, like,
years ago.
Started out as a little bubble and then filled itself. Like the
Unreal.
tissue just grew in.
Yeah, totally unreal.
How do you?
And so, yes.
Sorry, just
No,
maybe a
it's one-
basic question. How
No,
do
go
you
for it.
know that it's differentiated?
So you mean like different brain regions? Is that what you mean?
I guess. Yeah,
Differentiated?
yeah.
I mean, structurally. So you can either that something is separated by membranes, or you look and see histologically. So like cell on like a cross section on a microscope that it is different in either
Okay.
structure or function.
Okay.
But I mean, differentiated has like a very literal term depending on the field you're in. And I would say for something like this, they probably have a very specific thing they mean by the word
Yeah.
differentiated. So I don't want to speak to that because it could be wrong. But I would assume things where you check for structure and function
Yeah.
differences. So that's 2013. So this changes everything. So now I was like, great, let's go on the journey. And so then I looked up every mini brain publication since then. It's fine. And I grabbed some little highlights for you. It's got some little highlights.
Do tell.
Oh, I will. So in Singapore in 2016, we have not just a mini brain, but a mini midbrain. So being able to make this version, but that is of a midbrain. And keep in mind that the midbrain is a really important thing anatomically and physiologically because we're talking like auditory and eye movements, vision, body movements, all this good stuff. The other thing that the midbrain has are dopaminergic neurons. So basically neurons that produce dopamine. Okay? And so that's important when we start thinking about the idea of diseases like Parkinson's.
Yeah.
And so what they did in this study, the Singapore team, is not only did they detect functionally active dopaminergic neurons, but also saw the first time documenting there's this black pigment called neuromelanin. Makes sense, right? Break the word down. And that's in that organoid model. That's the first time I've ever seen it in a model. And the thing is, Parkinson's happens when you have those low levels of dopamine, but also you see a dramatic reduction in that neuromelanin. And that's the thing that actually causes the tremors for Parkinson's. So I also saw...
Why does it cause what do we know? Do we just...
Why a
is it like
reduction in neuromelanin production does that?
Is it just correlational, or do we know what its function is to... Like, does it prevent damage the same way melanin in our skin does in some way?
Right. I don't know that. It's one of those that's implicated in. So
got it.
Cause risk correlated. I don't know. But neuromelanin is one of those things that I wasn't even on my radar, hadn't heard of until I was just generally looking at Parkinson's and saw it come up, but I haven't followed that trail yet. Great question. Within this study, I saw this really, casual mention, these brain organoids, so these mini brains we're talking about, how big do you think we're talking? Like, if you ballpark.
I mean, I'm imagining 3-inch mini brains.
Yeah, I mean, like when I ballpark it in my head, it's like an Oreo.
Yeah,
but I feel like the real answer has probably got to be smaller than that. That's
I want just
like
legitimately the image that your brain conjured.
I was also thinking of food. It's like, these sound like little hors d'oeuvres almost.
I'm not going to rule it out.
I was thinking like pea-sized, like smaller.
Mini
centimeter-ish.
Oh,
sure.
Okay.
All right, so we're all here, and I had something similar actually because when I was thinking of differentiation and actually having different regional, like sections, I was thinking of like Petri dish, like maybe a centimeter, couple, 2 to 3 millimeters long.
Very mini.
That's real tiny.
That's so small.
You can't eat that. There's no calories there.
Yeah.
You don't know.
How many silicon atoms wide is that?
I only
42.
know.
Ish.
Dang, that's
30 minutes or less. So that's really incredible. But again, here's this whole thing where it's like when we talk about models, all models are wrong, some are useful. That's like one of those things.
Sure.
So with human disease, especially neurodegenerative things with the brain, mice can only go so far. Animal models can only go so far.
I mean, I guess maybe you're getting there, but like, have there been successful studies or whatever research on these mini brains that has led to a more deeper understanding of larger actual human brains?
2 parts to that answer. First, yes, and I'm about to very quickly just give you a couple. And then 2, these are actual human.
That's the part I
brain.
don't
like that. That's why this is incredible. Is that you're able to manipulate and look at what disease does and the pathology within this thing.
Okay.
I mean, it's human cells. Some of these teams made them from skin cells that then they induced, which is
that's weird. Not bone marrow. I feel like that's bizarre.
Amazing, right?
The fact that you can make your skin cells go back.
A skin cell should not become a brain.
No,
to be fair,
you should not induce pluripotency in it. That seems weird to me.
There's stuff about like using skins or just like maybe it's just stem cells in general, but maybe skin. I thought it was skin cells. Skin cells.
They have way less fat than the normal.
But to like revert them to stem cells and then grow organs for like transplants, something like that.
Yeah.
So this is like, I mean, you can't really transplant
it's a thing
brain
that happens.
as far
Skin
as
cells
I know,
have been used, which is just, but it's just weird that like skin, like what? Why? Yeah, it's amazing. So February of this year, Brown University in 2015, so 2 years after that first one, they start making mini brains, but like way cheaper and way faster. So they basically published a paper saying, hey guys, we're doing it, but here's a better way to do it. So that was in 2015. The same team, though, in February of this year, they actually, oopsies, didn't notice this little something called, after about 3 days of culture, about 2/3 of all the mini brains had actually grown networks of non-neural tissue. And when they looked at it, they noticed that it was actually like self-assembled, so just happened on its own. These tubes made of cells and protein that are actually found in blood vessels. So vasculature.
Weird.
They basically started growing vasculature.
They're trying to escape.
Well, the thing is, there's of course no blood.
Yeah.
Blood comes from. Well, that's the thing. So here's the idea now: is oh my gosh, that means that we could make a mechanized heart to then feed it. And then we
Oh,
can
God.
study things about brain disease that have to do with
It's gonna be alive.
vasculature.
Yeah.
Well, but I mean, like, that's amazing, though, because think about strokes, think about things like brain injuries, strokes, or Alzheimer's, diseases like diabetes, because a vasculature is not just blood. Think about what's in the blood. So things like glucose and also oxygen in these things. So please don't hate me. But I wrote down to myself, here's the mini-ception. Mini brains make mini veins. This is where I do a mic drop, but I don't want to drop this. It's fine.
But it would go nice on like a little cross stitch that you hang above the
I mean,
door
it's
the
in my
lab,
kitchen
like-
right now.
Yeah.
Actually, like right above the oven under at home is where the mini brain making mini veins of a heart lives, is something like that. Yeah. And then July of 2017, we had a team at Yale, and they wound up making 2 different kinds of mini brains, one that produces inhibitory neurons and one that produces excitatory neurons. The thing is, when you don't have a balance between that inhibit and exciting neuron balance, you have issues that are implicated in like autism and schizophrenia. And so basically they grew both and then fused them and then track the migration of the inhibition, which is just very cool. So that's applied directly to
bonkers.
see that.
Just
Are
crazy.
these so there's no blood flowing through these brains, but are there?
Nope.
Is there like?
But
are
there's
these
the
neurons
infrastructure.
firing in some way. Like, are they producing the neurotransmitters and they're
Are they producing
shuttling
electricity,
around?
right? So
Like,
one of the ones I saw said that they were producing electricity.
huh.
They're like a nutrient soup. So
yes,
they're
the
being
gel.
grown inside.
Okay.
I've never done any 3D scaffolding work, but
But you're exactly right in terms of that's the basis.
Yeah.
so they don't have blood flowing around them, but they have all
A
the
medium.
ingredients they need. And
So it's like Krang.
what's that?
What? What is that?
Tuna's with me
From
from Teenage Mutant Ninja Turtles, guys.
I don't
He's the
guy in the
know.
stomach?
He's a villain, and he's just like a brain in this big like dude suit, and the thorax is like transparent, and you can see the brain,
in a fluid.
which also has eyes and a mouth and little arms. and
Now
he's
there's
just in a
miniception.
fluid.
Yeah,
okay.
So
then yes.
okay, now
That's
I'm
fair.
here. I get it.
Yeah.
It
You're
all
here.
makes sense to me.
Yeah.
And that's how they turn it into brain tissue too, right? That's
yeah,
how you
those
take
are growing in that medium.
you take the stem cells and then you put in a bunch of growth factors that make them want
Exactly.
to differentiate. Differentiate in this case meaning turn into a different
Yeah.
cell.
a skin cell versus a neuron. Yeah.
Okay.
So you dump stuff in the medium. I'm sure they have a whole process where they like put
Yes.
some in, wash it out, put a different thing in, wash it out. Eventually it becomes a neuron and then it has all the
Yeah.
stuff.
And that's what all these
it
teams are
needs.
doing now, is saying who can do it like quicker and cheaper and more efficiently. And that's the study from 2 weeks ago, which was from 1 week ago, which was my original search, which found basically, hey, we can make mini brains. The thing is, a lot of the protocols are inefficient, or they're really different, or there's variability in their ability to get the architecture together in 3D. La-di-da. We have a way that's simple, it's efficient, and they are reporting differentiation method in which 80 to 90% of structures expressed the markers that were characteristic of the neuroepithelial organization. So basically just a week ago, the big one, and they wound up testing it with Zika and virus, looking at that. So this is an ongoing field.
Yeah.
The mini brain thing, first time reported.
Next week they'll try it with pumpkin spice.
Right, it's fine. It's fine. First reported in 2013, but now everyone's working on it, not only to make them quicker and cheaper, but also to see and use them and induce disease, basically,
Yeah.
to be able to then study it, et cetera.
I guess the thing that freaks me out about it is like, how do we know it's not conscious? And I don't know if that's just because I don't have a good understanding of this.
No, that's a
But, like-
philosophical question. I don't think it's wrong to
Like,
to
can
ask
we
it,
throw
but
it in an fMRI machine and show it pictures of love and French fries and see if it like responds or
responding to something is different than being
sure,
conscious.
yeah,
So I'm saying like that winds up being a question on a much deeper, like almost
too deep.
it's too deep. It's
Mhm.
you deep, not many. Yeah, it's
Oh, man.
it's a valid and great question. It's just not. I think it's not like a universally answerable.
Yeah.
because I think that that's in the realm of philosophy.
Sure.
Holy fucking science.
Holy
Yeah.
fucking science. Stefan, Ceri, would it be bad form if we double dipped and did a psych episode on that? Because it sounds pretty sweet.
On mini
Oh.
brains?
Many brains.
Yeah.
There's a ton of study. I only picked out a couple of like what I thought was, hey, this disease, this application, mini brains, mini veins, check. Yeah.
Right.
Can I come up with a cool
There's like
tagline?
so
Right.
much, yeah.
I thought you were going to ask us to double dip in like barbecue sauce, and I was like ready to be.
Is everyone
really
starving? Like,
grossed
what's
out.
happening?
Tuna didn't eat his half bagel.
I know.
honey.
I missed out on my half bagel.
Can I grab a second?
Yeah.
Yeah. Oh, because it's probably about that time,
The
isn't it?
sponsorship.
Yeah.
Yeah. Oh, yeah. Let's
Yeah,
do a sponsorship. Break them
Hashtag
open,
spawn.
people. We are here at Holy Fucking Science. We are sponsored by Big Sky Brewing, makers of Moose Drool Brown Ale, right here in Missoula, Montana. Largest brewery in Montana, available in many of the great states of the United States.
Check Ray McNally.
Very tasty, very tasty.
Good.
That went well, I think.
Yeah, it was a, you know,
Proud
a
of
little
you.
10-second spot.
Yeah, you gotta do it. You gotta. Thanks,
Oh, wait,
Stefan.
we gotta
Back
do
to
a-
you, Stefan.
Back to you. Or us. Tuna.
Is it me next? Or?
Thank
Right?
you
Oh,
guys.
I think so.
I played
that
mini
was really
golf
cool.
Oh,
more
right.
Well, no, I think it's my turn next. I think Stefan.
Okay.
I don't know.
Because
Oh, did I get it wrong?
Oh, yeah, yeah.
06.
Sorry.
11.
Yeah.
Sorry.
The year
My
after I
bad.
graduated high school. So
Oh,
now I'll always
yeah.
remember that.
Just
Because it's mini golf.
That was the year you graduated high school?
No,
Oh,
year
Tuna-
after Tuna graduated high school because it was before he got
Oh, that
friends,
was his-
remember?
Right. They all moved away.
no friends
to
to
here,
know.
actually. Jerks.
Now you're here.
Yeah,
now
we can
you're
reach
I
the
know.
levels.
It took me a few extra years, but I made my way over.
Good. Okay. I also was confused about the miniatures prompt.
my girl.
My first thing was D&D, and I was like, how do I do minifigs? Like there aren't that many things that are smaller versions, and I didn't want to do like mini horses. So instead,
Little Sebastian.
yeah, I'm just gonna sing the Little Sebastian song for Parks and Rec. So I just went with something small because I did cell bio, and so this is easy for me to talk about, and I can answer questions, which is always my biggest fear on Holy Fucking Science.
Good word.
So symbioses are a really important part of nature, and you've probably heard of them before. That's like Finding Nemo, clownfish, and anemone. That's an example of a symbiosis. But there's also endosymbioses, which are where one organism lives inside another. So this is like an anglerfish, if you have bioluminescent bacteria that live inside the little thing, and then they mutually benefit from that relationship. So the bacteria usually produce food or some other nutrient that the organism needs, and the organism produces waste or something else that the bacterium needs. And in 2011, scientists discovered the first endosymbiotes inside a vertebrate that wasn't parasitic.
Prove it.
So, like
Every
malaria
endosymbiont is
or toxoplasmosis, those are technically endosymbiotes by some degree, but they're parasites. They feed off of their hosts. And most endosymbiotic relationships are with invertebrates because their immune systems aren't as developed as vertebrates. And so, like, if something invades our body, we're going to fight it off. And there's a very low chance that something that shouldn't normally be in our cells is going to stick around. But that's not the case with a species of green algae that lives inside the cells of spotted salamanders.
holy fucking science. I love that.
Yeah.
Ooh, is it a digestion thing?
We're not sure.
Okay.
I will take the same approach Brit did and like step through the research
Yeah.
and how we discovered these things. Nutrient related things is our best guess, but mostly we're just not sure how algae is living inside the cells of this dang salamander because it's very weird.
So inside the cells, you're
Yeah.
inside the salamander, or literally
Inside the cells.
Oh.
Of a
So the
salamander.
algae is small enough that its cell is
Its cell is
been engulfed with
That's like
phagocytosis.
it's like mitochondria.
I was just about to
Yes.
say it's the endosymbiont
Yes.
theorem. Yeah,
it is like mitochondria,
and
but
chloroplast. Yeah,
and chloroplast, but just like a tiny algae cell floating around in a slightly bigger.
is it unicellular?
I'm not
Algae?
sure.
I feel like that'd be really important, right? Because
Yeah, it probably is unicellular,
I
but
want-
collects in
wait, is
bigger
there
masses?
unicellular algae?
I don't know. It's a green algae. I don't know if that matters because there's red algae and other types. The species is called Oophila amblistomatis, which basically means ambystoma is the salamander. Oh, of course you know that.
Tiger salamanders are some of my favorite things.
So you know their science good.
Now you got two favorite things.
Yeah. So ambystoma and Oophila basically means egg-loving, and that's where we found the algae in the first place. So we thought it worked like, I think in several species of salamander and frog, I didn't look them up, so fact check this, podcast listeners. There is algae that floats in the pond near the eggs, and sometimes they pass through the egg membrane and just kind of like chill in there with the embryo. And what happens is the embryos spit out waste products, which are usually really nitrogen rich. So in the same way, like bat poop is really nitrogen rich. And the algae loves nitrogen and needs that as a nutrient. And in exchange, the algae photosynthesizes, takes in nutrients, and releases oxygen, because that's photosynthesis. You take a bunch of stuff.
Fucking
yeah.
But it's doing this within the cell.
So first we thought it was just within an egg. So which I guess is a type of cell, but like within the growing egg, embryo separate from this. But then in 2011, so hundreds of years, we knew that frogs and salamanders lived near algae, and there's probably something going on there mutualistically. 2011, we found that there was actually algae inside the embryos, and like somehow during development, they work their way into the embryo. We're not sure the process. We don't know the mechanism at all, but it was just like studying them in culture. They like tagged the algae with a fluorescent protein and saw it floating in the egg outside, and then saw it floating inside the embryo. Yes, Brit.
I'm raising my hand for those of you that are just listening, because I understand I have a tendency to interrupt, which I promise is out of just excitement and not out of being horrible. But I still recognize that I do it. So I figured I would raise my hand because I'm very excited right now. Is it fair to say, though, that because mechanism, et cetera, is unknown, that it is in fact unfair to say that this is not parasitic? I find it hard to, like, the reason why when you first said it, I was like, prove it. Because truly, like, I find a hard time thinking about this and not seeing a gain. I understand that parasitic means that there's gain and then loss, like, whereas it can be mutualism, right, where we get remora shark, et cetera. When there's so many unknowns, I don't know. I would venture to say that parasitism, I mean, even because if they're eating something, some piece, maybe there's a protein or something within that cell, and then the cell's not actually getting anything, then I guess for parasitism there need to be a harm. But it's but they said there's no harm shown as of yet, I guess.
There's no harm shown as of yet. So we know more now than we did in 2011. There was a study released in May of this year where we looked at the messenger RNA in the cells of, they did two comparisons. The salamander cells with and without algae. Because apparently they're in most cells, apparently not all the cells.
In the cells.
And so they did a comparison of what messenger RNA was expressed in both of those, and the messenger RNA of algae within cells and outside of cells. Awesome. Basically, what messenger RNA tells us is the types of proteins that are being expressed and the types of genes that are being expressed, because it's within that whole translation of what's encoded in the DNA to what proteins are produced. And so it basically tells us what the cells are reacting to and whether they're stressed out or whether they're working normally, etc. Does that kind of make sense?
Yeah.
I got excited, so I'm getting deep into the cell bio.
It's so good. The way I explained mRNA was that DNA is like the older sister that wants to stay home and doesn't want to leave the nucleus. And they're like, oh, I don't want to run that errand. You go. I teach with a lot of analogies when it gets like cell level. So mRNA, I just imagine this like bratty little sister that's like, oh, okay, fine. I'll take it over to the ribosome. God, you're so annoying. I know you'll tell on me if I don't. So they will leave.
Yeah, they'll leave the nucleus.
Leave the nucleus.
Helpful. And so they learned that the algae is struggling really, really hard inside these cells. Like, it is not a favorable place for it to be. Inside an egg, it's fine because there's enough, like it has the ingredients it needs for photosynthesis. But within a cell, the algae cell is very, very stressed out.
Oxidative stress, like is that how they're measuring that, or just on?
It is producing heat shock proteins.
No shit.
And autophagy related proteins. So it's basically like, should I die? Should I not?
Also takes way more smoke breaks.
Yeah.
You should see the state of her hair. Could just like falling out.
Not doing well. Not pretty. And instead of photosynthesizing, which is like what algae would like to do, is photosynthesize, produce oxygen and glucose. Mostly what it's doing is fermentation because it doesn't have...
It goes anaerobic? What?
Yeah, it doesn't have the ingredients.
Sorry.
No, I love how much you're excited about this because I'm afraid of losing people, but the fact that you're following all this is just good. So yeah, it's consuming starch and glucose and producing things like, I don't know, I didn't look into it, whether it's ethanol or lactic acid or formate or acetate, one of those other compounds.
Pathways
for anaerobic. It's taking a different pathway to produce energy because it's so nutrient deprived within a salamander cell. And then on the flip side of that, the salamander cells themselves are showing no stress response whatsoever.
Bye, we good.
Yeah, it's just like, you're going to make a home in me? That's fine. You can chill, little algae.
Is this like a fourth kind of symbiosis? Do you know what I mean? Like it invaded, but then it's being affected
like,
negatively.
Oh, shit.
This is like anti-parasitism. You know what I mean?
Yeah,
like,
it's not getting... We're not sure what the algae is getting out of this.
But it's able to survive.
But it's able to survive.
But not in a good way, because keep in mind, when something undergoes fermentation, when it has the option of aerobic respiration, it is making way less ATP. Like, this is not the ideal. This is like its shitty version of like, guess we'll have to do fermentation.
Does it have the option if it's within the cells that are internal to the body?
Yeah, I don't know. I mean, this is all like so beyond anything I've heard of.
Strange.
No, it's like way cool, but I'm wondering, like, it's almost like someone screwed up. Someone screwed up. You know what I mean? Like, and
this algae made some bad choices to get where it is today.
Right. There's issues. There's a past. It's checkered. It's fine. So but I have to wonder now, how long has this been going on? Because potentially this is maladaptive and it simply hasn't died off. The deleterious mutation that has made this be a thing, right? It's just simply there hasn't been enough selective pressure for it to be like, hey, nice trying the creativity, but that was a horrible idea. And that's truly, that happens a lot. Like, we see instances of things that are just like crazy, but then it's just we're seeing a snapshot and it actually is maladaptive and it is not going to persist, kind of thing.
We've known about
this is so cool,
like algae related to amphibian eggs for I think 2 centuries ish. So since early 1800s, I think naturalists were observing that. We only confirmed that the algae entered the cell in 2011. So I don't know if this is a relationship that has been evolving over the past couple hundred years, longer than that or not.
Right.
But it's definitely to a point where both the algae and the salamander have changed their phenotypic expression, like what is happening with their cells. Like the salamander has suppressed genes that would normally mount an immune response and freak out because of the algae. And it's just like, if this is here, it's not hurting me.
Right, either habituation or something is beneficial that we just simply don't know.
Yeah, so it could be using nutrients that the algae is producing in its fermentation process, but we don't know. And so the next thing that they're going to look into,
is the salamander just drunk all day? That's its benefit? And it just wants to get down so it reproduces more?
I love everything about this.
Yes. That's it. I think you cracked the code.
Because amphibians...
What's the phone number of these researchers? I've got ideas.
Salamanders drunk. They could pass down. We're not sure if the algae can be passed down through eggs, instead of just being absorbed from the environment. So they could like mate and then pass on algae to their babies, and then like drunk babies. Don't drink and drive, kids.
Hashtag drunk babies for the children.
Yes.
For the children.
Hashtag HFS, hashtag drunk babies, hashtag for the children. Really important point there being the amphibian thing. We all, I think, had a mammal-centric idea of this whole in the eggs like inside of the we just think of eggs or even just things inside of the body or inside of cells not being totally permeable to the outside environment, right? And that's natural because like we are not amphibians.
I
Tell me about
because I think about chicken eggs. Is actually the first kind of egg that I think about.
Those are definitely not amphibians.
Well,
I think of
but also
tapioca
permeable.
Good.
Right?
Yeah. Okay.
Boba.
Semi-permeable.
Right. So I'm jumping from eggs over to just amphibians with skin. Like I mean, they are semi-permeable to their entire environment. So I'm saying like from a mammal perspective, like it's easier to forget something like algae or something in the environment. And that's why amphibians are at so much risk to pollution and things because like in the water, I mean, it's not like I eat it and then I have a lined, system that it is. No, the skin itself is semi-permeable. And so that idea of symbiosis happening with an amphibian species is a totally different mechanism from like the mammal thought of this. And I'm saying me too. Like I was forgetting like the idea of, oh wait, yeah, that's right. The algae's just right there. Unbelievable. Yeah, that was way too cool.
Have they observed because I assume the algae's life span is shorter than the salamanders. So it's like
Probably.
It is successfully reproducing inside of the cells.
Oh, interesting. I didn't look into that, but I would guess.
I'm going to spend the next 4 days learning everything about this. I'm so obsessed. So I'll send you an email.
Okay, cool.
Yeah. I'm not entirely sure how algae reproduce. They bud, maybe, or
spores.
Yeah.
Question mark.
Well, when a mommy algae and a daddy algae love each other very much. No, that's as far as I go. Yeah. No,
Tell us, Tuna.
I never heard the rest of the story. I was hoping you could tell me.
Maybe when you're older.
Protect the children's.
For the children. Green algae 6 is what I'm
Yes.
going to type in. No safe search, green algae 6.
Yeah, I don't know if they've studied them long enough either, or if it's just like we picked up a salamander that we know has algae in it. And then
that's kind of the thing I was wondering too. Is like, did they study the same salamander at different points in its life cycle to see like if they are slowly like dying off, and then by old age they have none? Brit has found something.
So. Algae sex?
There's something called a crampon.
What?
Isn't that like
the climbing?
The climbing thing, right?
I don't know.
Wait, what?
There's a spiky climbing thing.
Yeah.
That's a crampon.
It's a crampon.
Right.
Yeah.
Yeah, so green algae both does sexual reproduction and asexual reproduction. I just had not heard crampon, and I got very thrilled.
But
That's
is it
all.
related to algae reproduction?
This is green algae. Yeah, because
What is
the crampon is actually a piece like of the stalk, basically, of the green algae, and so it's following the crampon through the process to like the new colony. The crampon is almost like the base.
So what the hell is the green algae doing climbing on stuff?
It's not tuna. Maybe we got it from the algae. Maybe the crampon has existed there the whole time, and we ripped it off, so we owe them money.
I'm just saying shoe crampon has way better marketing than algae crampon.
What if shoe crampons are just made of algae? Just like really slippery.
Now we're thinking. You are onto something, my friend.
Yeah.
Wow.
So yeah, they do both, but liberation of spores is a phrase. So spores is
Yeah.
Oh, yeah.
I figure like
Okay.
it's a plant.
it's gametes. It's damn gametes, right?
Yeah.
But it's liberation of the spores? Is how they phrase it?
Liberation.
That
of
sounds
the
like they're
spores.
like breaking them out of like, oh, we're going to algae jail and we're going to liberate the spores.
I'm in.
Man. Yeah.
I mean, really, aren't we all just waiting to be
Liberated.
liberated?
I just want my spores to be liberated. That's all I dream of.
Yeah.
You know?
Yeah, there's a
Do
lot
that
going
at
on
home.
here. It's very exciting.
Man. Holy fucking science.
Yeah, that was cool.
Yeah.
Holy fucking tiger salamanders are so cute.
We should put mini brains in the cells.
What does that even mean? I understand that you're still concerned, but that actually doesn't mean anything.
We could take like there's a really big
2
bacterial
millimeters?
cell.
That's bigger than a cell. What shit do you mean put it in a cell?
There's like
Yeah,
the deep
make 'em
sea cell
smaller.
that's huge.
Miniaturize.
Right? I forget-
Xenophyophores?
That thing. Yeah. We did a SciShow on it. I didn't edit that script, so I know nothing about
okay.
them. But There are cells big enough that could fit a mini brain.
Yeah, there's cells big enough, Brit.
Yellow Couch was having its own conversation.
This is why Stefan wants to know if they're intelligent, is because it's like you slap a little pair of eyes on it, mini eyes, and then you can just ask them questions later and be like,
Can
hey,
we put googly eyes on the mini
yeah,
brain?
what is that
That's what
algae doing
Google's
in
from.
there?
Yeah.
You just ask questions to googly-eyed mini brains. We thought it was from Googleplex. No.
Far be it.
Well, we learn
boy.
something
There's a
every
not
day.
so mini brain at the center of Google headquarters, like deep underground in a vault.
2 to 3 millimeters in Seattle somewhere.
Yeah.
Oh.
They protect it with their life. That's why they're all this. Non-disclosure.
This is a conspiracy theory I can get behind.
Yeah.
Oh, yeah. I mean, this is what Minority Report was based on, right?
Crampons,
Like,
yes.
Yeah.
Crampons.
That was, crampons are those weird orb things that the Minority Report thing.
It's those
puked
climbing
out.
shoes made of algae. It's fine. TM.
Okay,
No.
so cool. Let's
Tuna.
go into
Okay,
it.
so it's me. All right. I'm going to talk about stuff all over time and space. So get ready for it.
All over
Yeah,
time and space.
I know. Well,
I'm
first we're going
ready.
to start with time. So,
Okay.
yeah.
There's-
Oh, time
Mini
and
time?
space, Brit. It's great.
Oh, one at a time.
Well, it is going to be one at a time, pretty
Okay.
much. Pretty much.
Great.
But okay, so we're all here in this beautiful place called Missoula, Montana. This smells funny sometimes. That fact is unrelated to this, but I want to make it known that this place smells real funny sometimes.
What does that mean?
Side fact.
Have you never been on the north side in the summer? It smells awful.
Is it the semen trees?
I don't know what
Ooh,
it is,
yeah.
dude. It's
This
awful.
was an episode we did.
It's bad.
I pitched it. I'm so glad I finally made it through.
Nice.
We didn't leave our house this summer because of
'Cause
respiratory distress.
Right. Okay, yeah, this summer it was
Next summer I'll
it
be on
had
the
different
lookout.
problems, yeah. But okay, but at the end of the last ice age, which was a little while ago.
Sure.
You had this thing coming down from Canada and covering part of Montana, like the very north of the US Pacific Northwest area, called the Cordilleran ice sheet. I have not said that out loud yet, so I hope I pronounced it
Fine,
right.
I believe in you.
Mm-hmm.
People will yell about it later.
Yeah,
It's
but I
great.
mean, big glacier, and right here where we're at was called Lake Missoula. It was the giant glacial lake.
Mm-hmm.
There's some art guy in the community who built a 3D VR simulation of it you can tour, and he put in little woolly mammoths and stuff. I don't know how scientifically accurate it was, but it was kind of fun. But giant lake. And the way we know that is because geologists went out and surveyed this. And at the time they did all this and found all this was early part of the 20th century. The way they did that was just going out and standing on top of a mountain and doing their surveying. And it was not incredibly high tech by any means. They had, a plane and a little viewfinder thing that they could see numbers on a stick pretty far away.
Sure.
And and so it was kind of a pain. And that was the only way you could build a map, a topographical map of these areas. And when they did that, first there was a guy, well, not first, but the important guy is J. Harlan Bretz.
Oh.
And he was a geologist from Michigan, but he did a ton of research in the Pacific Northwest, specifically in the scablands in eastern Washington and Oregon.
Mm-hmm.
And that's where he noticed erosion patterns, weird erosion patterns, out there. He spent 7 years of his life just traveling and surveying all this stuff. And he noticed, when he was out there, all this weird erosion. And it just kept gnawing at him as he was doing this. And eventually he figured out it was flooding. Some giant flood happened and left behind all this crazy erosion. And when he brought that back to the U.S. Geological Society, they told him he was a dipshit. Because they were like, no.
Quote.
Yeah, right, yeah.
Subtext.
We
Man,
talked to
huge
Rand
dipshit.
McNally, and his maps don't show
It's
this
like,
at all. You
yeah.
dipshit.
Yeah.
Who Rand McNally makes maps? I just thought the entire time. It was like a pop star.
Right.
Oh boy.
Well, so he catches a bunch of shit for this because it was like, what did they call it? It was the difference between like, I forget the two terms. Basically, there was the view that was prevalent at the time that like geology happened slowly, and it was like basically happening the same way now as it always happened, and everything we have is just like that, those slow changes building up.
Yeah.
And he was like, no, there was like something catastrophic happened, and the Geological Society would not have any of it until there was a geologist from Montana called J.T. Pardee, and he worked for the United States Geological Survey. So he had less leeway to like fuck around because I guess the USGS kept a tight leash on their geologists. I don't
still
know.
do.
Do they?
I don't know.
The shortest leashes in the federal government belong to the Geological Survey. That's a fact.
Is it? Are you fucking with
No
me?
one's on a leash.
I
Tuna.
don't know, dude. I don't know. Well, the way I read about it, it was like he couldn't outright support Brett's because it was like
There are
he was a
issues when
governmental
Yeah.
employee for this, like whatever it was, there was like politics at play.
Yeah.
But his research was all about Lake Missoula. And so he had noticed, like once he got a look at Brett's work, he said, oh, that would make sense. And together they figured out that it was Lake Missoula and there was just an ice dam and that broke apart
Oh.
and
Yay,
sent this
together.
huge flood from Missoula all the way out to the coast and left this like swath of destruction. And Pardee ended up tracing back, like he could find like, oh, the giant boulders that had gotten
Yeah.
pushed and like all this crazy thing. And even now, like as once that was accepted and research has gone on, they found evidence of like 40 different cycles of this like ice builds up,
Oh.
fills with water, floods,
That's cool.
and then keeps happening. Yeah, just like all this crazy shit. But they had to figure that out by standing on the top of a mountain and measuring stuff with a stick that they can look through at another stick. Or at least I didn't do very well in surveying class, so maybe I understand it a little wonky. But okay, so now that was our time hop.
Oh.
What?
Yeah,
What's many?
we'll get there. Because now
Tuna,
I'm going to
you
take
tease.
you. I know, yeah.
You're
You gotta
bad.
wait. You gotta
okay.
wait, Brit.
All right.
Can't jump the gun.
Oh, I always do.
So now we're going to jump through space, and we're going to go to the Mosquitia region of Honduras.
Great.
Yeah.
See you there.
I'm
I'm
bad
transported.
at- yes.
We're there.
So basically, it's this area of Honduras, like a big chunk of Honduras is really intense jungles that you can't really get through. There's like all sorts of deadly snakes and spiders. And
Sure.
That's the reason I want to go to
oh
Honduras.
yeah, I mean it sounds intense, but there's been this legend of the White City. It's called, and I don't know how to pronounce. It's La Ciudad Blanca. I don't know what accent that was,
Sounds
and I definitely
Ciudad.
didn't. Is that
I think
that?
Ciudad, yeah. is
Ciudad?
that-
Yeah, that's.
Yeah.
I still am
Yes,
not
high school
saying it
Spanish.
right. Oh, right. So that's been this myth, and that's hundreds of years. Like since the conquistadors got there, they heard tales of the White City, and it's just a legend built up around a bunch of other like legends. But what happened is that in 2012, a group of archaeologists and anthropologists got together and they managed to rent. I don't know if it's rent. They worked out an agreement with both the Honduran government and with NASA, and I think there was a Canadian agency involved with this too. But they used LIDAR
Nice.
to map this big chunk
Nice.
of Honduras. And when they did that, they found like a massive civilization that was in like this perfect little spot and would have been an amazing trading post. And it's something that for hundreds of years, people died searching for it. And granted, it's not like the White City. It's like the real version of that, which was like this city. And they found like a couple of other things. But it was a huge plane. They had to like secretly take it out of JPL and take it down to Honduras. And it was like this janky little plane. There's a book that came out about it by Douglas Preston called The City of the Monkey God. And he talks about being crammed in the back of this plane because they built it for the pilot and the guy who was running the LIDAR and nobody else. But he would sit there, and it was just this crazy thing. They had to set up base stations because they need to know exactly where that plane is. So they have all these GPS systems going off and like LIDAR sweeping across, and they would do like a couple of miles a day or something like that. And it just took them forever, and it was millions of dollars to do this. And just crazy expensive, but huge discovery. So the miniature, what?
Have we been there?
Have we
On
been?
the ground?
Yeah. So that's part of the book. Preston's book was like he actually went on the expedition and went to the initial finding of like, now we have this LIDAR map that shows us exactly where it is and how to get there. We can get there now.
This is reminding me a lot of the movie Congo.
And we're back to Michael Crichton.
It all comes back to
Well
Michael
done,
Crichton.
everyone. Go team.
Right. So the miniaturization comes in because that was 2012 and LIDAR was crazy expensive. Like, again, they had it for like a couple of weeks, I think, and it cost them millions of dollars. Like, and it was a huge hassle. Like, they had to get
is for free now and available on Google Earth Engine.
Well, that's the thing is the miniaturization LIDAR has gotten so much cheaper.
Yeah.
And now even the Montana Bureau of Mines and Geology has been looking into getting their own drone with a LIDAR on it. And that's just like...
Sorry, that's one of the technologies that they're using for self-driving cars.
Yeah. And that's the thing is now it's becoming cheap enough, so it's not millions of dollars anymore. It's small enough and cheap enough that you can have self-driving cars. Or there's what got me interested in all of this is actually they just found a fault line by accident down in the Bitterroot.
Yes.
And it's like...
I read about this.
Yeah. And it's just something, it's been there for a long time, like fault lines are. But nobody's ever known. But then there was a geologist, Jeff Lawn, I think his name is. Yeah, Jeff Lawn. And he was doing floodplain analyses down south of like Hamilton. And covered a bunch of area with LIDAR, and he was given a talk, and the state seismologist, Mike Stickney, looked at his map in this PowerPoint, and he was like, that looks like a fault. I didn't know about that fault. And he went out and looked, and like, sure enough, there's a fault down there. And he says like, and this is a big one. Like, if it were to like slip or do its thing, it'd be probably a 6.5 to 7.5 earthquake. And nobody knew about that.
Yeah.
And that was just with like the one that Jeff Lawn got a hold of. And now they're thinking of buying their own and like looking into it. And it's crazy what you're going to be able to find with it. Like, I know in the Middle East, they've tracked like a bunch of trade routes through the desert because like that shows up on a LIDAR.
Yeah.
And I know like there's been talk of using it to find places where Lewis and Clark traveled, like finding their trail because you can look down. And I think, I can't remember where this one was, but somebody has actually used that where there was supposed to be some campsite. And it's been written about in a lot of historical things. And it was like, we can't find this thing anywhere, but it's supposed to be right next to this river until they get LIDAR and cover like a big swath of the river. And then it turns out, oh no, we can see where the river was flowing 150 years ago. And where the campsite was because the rivers moved. So, of course, our directions suck now.
Can we do it from space?
I think there is LIDAR from space, but I don't think it's as...
Not as maybe
fine.
the resolution.
NASA, that's one of their data sets,
It just fascinates me because now it's going to get even cheaper. Like, what happens when you can have LIDAR on your iPhone? Like, what are we going to find then?
We use, to me, it's not what do you find, but how can we use it to do problem solving? And we use it in conservation of wildlife populations actually quite a bit. A lot of this remote sensing data because the like number one cause of like species loss at this point is arguably habitat fragmentation or loss or conversion. And so not only like tracking of animal movements, so like telemetry, but actually habitat usage and how that is occurring, populations on the landscape in space and time. So those things are directly applicable to, global biodiversity loss, like much less like finding cool shit, which is still
Sure.
that's great.
Oh, yeah.
That is fabulous. But there's like real problem solving
Yeah.
to be
Yeah.
done with these kind of technologies. And as miniaturization happens, again, having it on your iPhone, to me, immediately I think of citizen science. And then having people in each state collect data on the movements of a wildlife population. I mean, you can keep going down the list, but
Yeah.
I mean, that really does allow the power of science in your hand kind of thing. And then what we do with that data, really amazing potential for problem solving.
Oh yeah, crazy cool stuff, and I love it. And it's like so many different applications because we're talking about practical applications. But apparently, there's been a video game already where it's like
More importantly, video game.
Well, this is how you know it's like, oh, okay, this shit's coming
That's fair.
out. Like,
Is that the Xbox Connect?
man,
Is it
I don't know
LIDAR
what
your
it was.
body?
I just
Oh,
said it's some
Jesus.
racing game, but they LIDAR'd a bunch of
You
race
race your
tracks.
self-driving cars.
Right, yeah.
Everyone loses.
Well, but it's very orderly in the meantime.
Thank God.
So they LiDAR the racetrack to like accurately model the tracks
Yeah.
and like the surface.
And it's like, yeah, it's down to like
That's
sub-millimeter,
down to the
like,
atom.
yeah, it's right.
What
they can-
kind of atom?
Couple silicon,
Silicon
mainly.
atoms.
But yeah, and the thing is, they did it with historic racetracks too,
Oh.
so that like ones that aren't in operation
Yeah.
anymore, and so now you can build
Huh.
like a 3-dimensional perfect
That's cool.
replica. I've seen a ton of cool
Yeah.
phototelemetry stuff, but not. That's literally just like going in and taking hundreds of pictures and then building everything yourself. I haven't seen it where it's like, oh, just like run a LiDAR in this space and then slap your textures on that.
We got some cool technology.
I know. We're gonna have some sweet video games about like
That's
tracking
what matters.
moose.
How good are
and,
the video
like,
games?
Tracking
gonna be
moose.
tracking moose in ancient glacial
Moose
lakes.
track. I mean,
Yeah.
deer hunting is
Important
a
things.
popular series of video games. So I don't know. Moose tracking.
Big buck hunter, perhaps.
Sure. I don't know what they're called.
I don't know.
There's a big game hunter. Is it Big Buck Hunter?
I
They're
think big game
both,
hunter.
both, all.
Probably, yeah.
All.
They're all knockoffs of each other, and you don't know
Right.
which
Yeah,
one's
yeah.
the worst at this point.
Brit's actually got a whole room in her house that's just an arcade.
You
full of
don't
killing
know.
things.
You did.
It's how I balance the conservation work I do during the day. I only do wildlife conservation research so that I can kill them all virtually downstairs. Fun
I
fact.
can better hunt these moose.
Fun fact.
I'm gonna save this
boy.
little bunny and then punch a cougar.
Yeah, I'm okay with that.
Yeah.
Cougar punching game.
Right,
I
yeah.
like it. All right. Well, that was great, guys. Thank you for joining me on this lovely episode of Holy Fucking Science. Very much for children. Behind the camera today, we've had Caitlin Hofmeister recording sound and running the
Yay.
camera, turning all the knobs and flipping switches and stuff. I have been Stefan Shin.
I am still Brit Garner.
I have been Tuna.
I've been Ceri and will continue to be Ceri.
And this has been
Holy Fucking Science.
Brought to you by Ram McNally.
Oh boy.